So I want to give a special thanks to Wally Dano for the kind invitation. We're very grateful. And also for Mr. Chris Rhodes, who actually made sure that we got here. Thank you. Thank you very much. It's been a busy time. So we're MedinCell. That we are. And I'm pushing the right button. I promise. Thank you. Our outline briefly. I'll talk a little bit about the corporation, what we do, about the MedinCell drug delivery system. This is a peptide conference. I'll discuss peptides. A lot of the data you will see is partnered data. It's cartooned, what we call. So just bear with me with that. There is a reason for that. It's confidentiality. We've recently gone into ophthalmology. I'll briefly review some of those slides and then give a commercialization update. Wow, that time went fast. Okay, this would have a map on it. It did. We are based in Montpellier, France. That's our headquarters. We also have a satellite office in San Diego, California. We are a private VC-free company. It's a unique term. We've been growing, and we are driven by entrepreneurs. We were founded in the lab of Professor Michel Vert in Montpellier University. And it's really important. Some of you may have heard of this company in the past. A lot has changed over time. Most importantly, I show in 2001, we filed the 5th generation of this technology. Earlier derivations of this technology had a di-block copolymer. With one polymer, you're not able to both control your burst and your duration. So I'll show you what we've changed to over time, but we're more than 10 years old now. Primary corporate activities are we design tailored formulations. Duration can be a few days, it depends on the indication, up to more than 6 months, even with a peptide. And we can, in general, control Cmax and duration together with this dual system that we have. We also pursue local delivery, so intra-articular, so into the knee, and also intravitreal and other eye applications as well. The eye is brand new, so I'll very briefly review that. We also have in-house proprietary programs of basically well-known compounds that we are deriving analogs of together with the drug delivery system for long-term delivery. And because we're a VC-free company, we pursue global health initiatives. We can do these programs internally without having a VC that's not happy with us, and we license these royalty-free. This is the system. It is soluble. So what's unique about this is that a soluble system. I did have a glass of water up here that I started with. I think somebody thought that was the other one. Yeah, can I get a glass of water? Just a little bit. Thank you, sir. But it is a clear system. This is not aqueous. It's biodegradable, biocompatible, and it's low cost. Of course, it's IP protected. We license this technology. But it's important to note that it's not a microparticle system. It's not a nanoparticle system. Because of that, there are less scale-up issues. So you're not trying to recreate a small particle size on large scale, which can be very difficult. This will just exhibit and show the system that we have. This is a vial of water being injected with the MedinCell system. We'll try that again because he's good at that. There you go. Thank you. And so what you see is this would be an injection into the body. This is just into water. And it forms an in situ hydrogel. So you've seen a soluble liquid, and it forms an in situ hydrogel, which is kind of neat. You'll see this get more white over time. What happens is the solvent is going out and water is coming in, forming the hydrogel. And this is what our system is. So it is a tri-block PLA, PEG, PLA, and a methoxy PLA. So it's a tri-block and di-block. That combination is what allows us to control both burst and duration. The original MedinCell technology did not allow that, and it did have injectability issues. So this is a big change from those days. A lot of things have changed. But what you see is that this turns into a in situ hydrogel, which can be a nice system because that is hydrolyzed over time. There are different levers that we pull in this tailoring of these formulations. We have a polymer library, so for the 2 compounds I mentioned, we have different chain lengths of the polylactic acid and the PEG. We vary that through that library. We vary the ratios between the two. We have different solvent systems I will briefly review. We also can do prodrug approaches, salt forms, but most of this is really varying the ratio between the two and the chain lengths. We have worked today with peptides on the order of 50 amino acids. I'm not sure if this is up to date. I think we've gone larger now. Proteins, as I said, this is some of our newer work, and we have extensive history in small molecules. Best small molecules that work for us are positive log D and hydrophobic. We can do other compounds, but that is what will give us the longest duration. Loading, 0 to 10% is optimal. We go up to 40%. Higher loading, you can sometimes have more injectability issues. I'm just going to show a variety of PK curves. This is not a peptide. This is just a traditional opiate. This opiate on the left side would be dosed. You see a nice strong peak here. That'd be half a day. You get a strong peak. You've got lots of side effects for that. And if you highlight and zoom into this area, which is the right side here, what you see happening here is the strong peak, the Cmax. The same dose formulated with the MedinCell system provides lowering of Cmax by a factor of 25 and extending the duration up to 10 days. So this is a half a day, it's now 10 days, but it's the same dose. Of course, it's above the minimum therapeutic level so that you actually have a positive effect. It's quite a versatile system. So what we do is we tailor design these formulations. In some cases, you may want once a week product and then going to once a month or even longer. Risperidone's a well-known antipsychotic, so this is something you might start oral dose, you might go to once a week and then go to longer period of time with tolerability. That's a nice thing to be able to do, so we do design this kind of program for various partners. But it can be very long. Many of these systems top out at 3 months, maybe a little longer, but we can do more than 6 months. I don't believe we've gone up to a year yet, but they can be quite long. This is a 6-month, this is an animal health product. We work in human health and in animal health. We also work with small peptides. So this is a comparison against a traditional PLGA product, which was a 3-month product, and this simply goes longer. It's sometimes a benefit. It depends on the target product profile of what the partner's looking to identify. But basically, this is the same cargo here. On the left side is an in vitro, and on the right side is an in vivo rat PK. This, some of you that are experts in this area, and I know there are a few in the room here, Chris, this still has a large Cmax. This is an unoptimized formulation. This is an older one, so I just want to be very straight and honest about that. We have very different formulations of this today. I can't present those, unfortunately. But this is a great therapeutic area, as everyone knows. Generally, compliance-wise, you once a day, once a week. One of the golden grails will be once a month. That's what this can do. Once a month of a GLP-1, that would be a nice, more attractive product for someone like me who is needle phobic. We also vary the solvents. So many of the peptide formulations that we do are in DMSO, not all. And we've now gone into proteins. Sometimes we use things like triacetin, tripropanin, and sometimes even a combination of tripropanin and triacetin. This is what we're doing with some of the protein formulations we are doing today. A lot of the proteins are happier in this kind of environment. Also for uses in some of the more delicate areas like the eye, this is a favorable system. So, but you do see differences in using just a different solvent system on the PK, which is sort of interesting. Can't tell my time, okay. Generally, we have good in vivo versus in vitro correlation. Not always. This is a good example where they're trending the same direction, but we will produce a number of formulations and then optimize them and do a PK and select from those. Sometimes you get an inversion of the PK, which is an unusual situation, but we do see that. But the duration of action is based on the partner. So it's based on the target product profile. What are you looking for? Is this a product that's a once a day, once a week, once a month? What can you do? What is the desire from a commercial standpoint for the final labeled product? Thank you. Compliance is everything in the medical field. And as I said, once a day, you might want a next step would be to once a week. So this is a nice peptide formulation that's once a week. I apologize that you can't really tell what the actives are, and that's intentional, but these are all real. That's one thing I can tell you. And we also apply this locally. And so this is an indication of an intra-articular delivery. So this is into the knee. These experiments were done in a sheep. We have a partnership with a group in Canada that's a group of physicians that do 8,000 knee replacements a year. They were interested in this for post-surgery inflammation. This is a case where you have a daily dose of one NSAID injected into the knee in this polymer system, lasting 3 months. So this is one daily dose lasting 3 months with a very low systemic level. So that's a nice system for that. We are doing that today with antibodies and small molecules. That's a very interesting program we have today. Regulatory considerations. This system is based on methoxy-PEG, PEG, and PLA, but they're joined by an ester bond. So these compounds are quite well known in the literature. They are all bound by USP monographs. Our system is a little different, so what we are doing is bridging these polymers with our safety package today. So it's very similar, but slightly different, and so that's what we're doing for that. A lot of companies have asked about commercialization. Gee, you have DMSO, you got some of these weird things. Who can do this? First of all, as far as a commercial GMP polymer partner, we have that identified. They are producing GMP batches with a DMF. And what's important is that also secures the trade secrets. And so there are a lot of trade secrets in the purification and the production of those polymers, and that's one of the things that we think are very important. Also, we have more than 6 fill-finish sites now identified, Europe, US, Canada, that can do this today. They don't need to do anything. They're able to produce this kind of thing today. Some of these formulations are aseptically filtered, some are terminally sterilized traditionally, and some are irradiated. We do optimize for injectability. This was not the case in the earlier MedinCell, but we do produce these candidate formulations, and then based on the target profile and the needle gauge, we figure out the force needed to express it. Our goal is less than 10 newtons, and we do optimize for that. It's very important for the final product. As far as intellectual property, we have the platform patent, we have the polymer trade secrets I mentioned, and we can do pharmaceutical composition patents on each product, and that gives us what we believe strong protection. I'm going a little fast, I apologize. We have finally, it took us some time, done the screening studies for safety in the eye. And what we have today is to express just today is looking at intravitreal. We've also done subconjunctival injections. This is not a thorough and complete slide yet, but what you see today is at least we are moving forward in this area. What we believe is a very interesting system is using tripropylene as an excipient, injecting in the back of the eye because we can do proteins in the back of the eye. That's an area that many cannot do. There are solid PLA systems and the like, but no one really is doing proteins that I'm aware of in the back of the eye. And so that's a very attractive area. Please contact us if you have any interest in that. What's our business model? This question has been asked a lot. Our business model is we have twofold. Traditional model where we do an FTE-based formulation development. We're very fast. We can do that rapidly because we have a lot of experience with that. That's the one model, and then we license that to the partner with a milestone and royalty-bearing license. Or we also work with a lot of virtual companies today. That model is more of a shared and profit model because virtual partners don't want to put a lot of money up. We understand that. We work in both scenarios. That IP that is non-severable to the platform we keep, that IP that might, for example, be in a formulation patent, that's licensed to the partner. And we keep the responsibility from the polymer scale-up. That protects the trade secrets. That's a very important part. And finally, we believe that this company is not just your traditional drug delivery company. We're well positioned to exploit the gold mine of drug delivery. Why? Because it's versatile. So we can work with small molecules, peptides, and proteins. We can do this quickly because we have a lot of experience with this library. It's scalable and low cost because it is a soluble system. And fill-finish sites are readily available. This is not an issue. So thank you for your time, and I look forward to questions. And I'll give you a demo as we're waiting for the questions.